Okay.
Have live stream.
Oh, Evelyn is not here yet. I think. Do you see if she's here? Roberto. Alina. I don't see Evelyn. Do you see her?
No.
Is she there? Is Evelyn there?
Yes, no, she's there.
Great.
I am.
Oh, great. Here.
Hi, is that Silvina?
Yes. You are on. I mean, you are for everywhere to listen to you. So let me introduce you the prize and you too, and then we will move on to your talks. So it is a great pleasure to present the first edition of the IUPAP Interdisciplinary Early Career Scientist Prize that corresponds to the years 2022 and 23, and to present the two awardees now connected on Zoom. Evelyn Tyne from Rice University. She got this prize for her development of new topological and geometrical analysis that reveal fundamental physics aspects which allow the characterization of robust emergent phenomena in complex systems, from quantum phases of matter to biological systems and the brain. So let me tell you a little bit about Evelyn. Evelyn joined the faculty in the Department of Physics and Astronomy and the Center for Theoretical Biologic Physics at Rice University in 2021. Previously, she was a group leader at the Max Planck Institute for Dynamics and Self-Organization, and before that, an African postdoctoral fellow at the University of Pennsylvania in the group of Danny Bassett. In 2015, she received her PhD in physics from MIT, where she worked with Xiaogan Wen on novel topological states in quantum electronic systems. She holds a Master in Philosophy from the University of Cambridge and a BS from Yale University. Evelyn is a recipient of several awards, including from NSF, Career, Dialogue, and the Chan Zuckerberg Foundation. She was previously a Simon Berkeley Research Fellow and Gates Cambridge Scholar. And let me introduce to you the other awardee, and then we move to the talks. So the other awardee is Stefano Martignani from New York University, and he got the prize for groundbreaking contributions to the understanding of the statistical mechanics of active and amorphous systems via the development of uniquely original approaches for quantifying order, entropy, and entropy production in systems far from equilibrium, including granular and active matter, neural networks, and biological systems. And Stefano received his Bachelor in Science from Imperial College London in 2012, followed by a Master in Philosophy in Scientific Computing in 2013, and PhD in chemistry from the University of Cambridge in 2017. He spent 2 years as a postdoctoral associate in the Center for Soft Matter Research, Department of Physics at NYU, and subsequently joined the University of Minnesota as an assistant professor of chemical engineering and materials science. Since 2022, he's an assistant professor of physics, chemistry, and mathematics at NYU. Prior awards include the Simons Foundation Faculty Fellowship, Gates Cambridge, and St. John's Benefactor Scholarships, and Outstanding PhD Thesis Prize from the University of Cambridge. He's an NSF and NIH Principal Investigator. And so let me stop my sharing. And we applaud them, of course. And so if you can turn on your, I don't know if I can choose them to be, let me see. Can I pin 2 people? I don't know how to do that. Anyway, so this is Stefano. Oh, I don't know what, there. So Stefano did get the medal. I don't know if you have it with you there. So that you
I do. I do. Would you like me to show it?
Yes, because we were going to have the award in which we would give you the medal.
The medal is there.
And Evelyn had, because we invited Stefano and Evelyn to come to China, and they couldn't do it, but we could ship the, at least for Stefano, the medal on time. So for him to show it and do this fake giving the award, this ceremony. And so we are pleased by this first edition and very pleased by the awardees. And so we can move maybe to the talk by Evelyn first and then by Stefano. And so Evelyn, if you want to share your presentation. And Evelyn, let me see, you're here.
Sure, thank you.
Sorry that you don't have the medal to show, but you will
Oh.
receive it on due time.
Yes, the anticipation is very high. Thank you. Thank you so much for this opportunity. I'm very grateful that even though I'm in the US, I can still speak to you and to really have this moment with all of you and see you face to face. We've been corresponding a lot, and yeah, I'm very excited about also even this whole category because interdisciplinary work is becoming so common these days, and I would love to tell you about some of these ideas in my work. And some of it is inspired by the fact that if you look at your brain network and you look at my brain network, they look different. They look like what you see on the screen here, sort of messy, entangled, blobby. Biology is often very heterogeneous and very stochastic. And I say this because as physicists, it's often a big challenge to us. How do we start to model these systems that are so heterogeneous and messy? And especially if we want these networks, for example, a brain network, these are neurons that are synapsing and connecting to many other neurons. If we want these networks to support robust function, so even though our brain networks look very different, if we were to look at them, yet we can support many similar functions. I am here speaking to you. We can support thoughts, language, communication. Our brain networks can regulate our bodies. And so a lot of robust function can continue. And I'm particularly interested in emergent function, so long time scale, long length scale behavior that is very stable, even though sometimes the networks often have much faster chemical reactions or much faster timescales that make up the network. So in circadian cycles, you have cellular reactions that are fast, or in memory, you have spiking timescales that are milliseconds, or sometimes even the lifetimes of cells themselves are shorter than the memories that persist. So how is it that we get such stable and emergent function, even despite the short timescales of the networks themselves? And as a theoretical physicist, I would love to think about how we can predict these global dynamics analytically. And this is a question that is really important in many areas of biology. So whether I mentioned the circadian rhythm, whether we talk about a global clock, or in gene replication, or even in large length scale and time scale that we need for motion, whether it's motion of a simple leech or in larger organisms like ourselves. So this is all examples of emergent function, in which we have many parts that act together to produce more than the sum of each part. And what I'm going to show you today is a framework, which is topology, to try to link this together. And I think that this is one way in which my work is, this example is showing this interdisciplinary work in which I'm bringing topology, very much developed in, let's say, originally condensed matter, but has been very powerful in predicting robust function and thinking about using that to predict these much more, let's say, messy looking networks in biology. How are these networks constructed? I'm going to show you one example. And these networks are often powered by the consumption of energy. So specifically chemical packets such as ATP or GTP. And this is one example in which we have a futile cycle, in which we have a phosphorylation, dephosphorylation reaction. And these were called futile cycles. You can see that people such as Hopfield, who got an amazing honor this week, as well as many other people have looked at these cycles, and they were given the name, these cycles were given the name futile cycles because they seem to go right back to where they were. And it's very, very, very intriguing for us as physicists also, because this is a very energy consuming function. There are continuous fluxes going from one side of this network to the other. This is not in detail balance. So this is very energy consuming. And yet it seems as if this cycle is futile, it's useless. And yet this kinds of cycle shows up in many different places in biology, metabolism, sensory systems, etc. And this is definitely very intriguing for us as physicists. We think that energy should be used in an efficient manner. That's often our hypothesis. And so here, my hypothesis is that these seemingly wasteful motifs, for which we don't quite understand why they exist, can contribute to system robustness, to stabilize the system. So with that, what I want to do today is to give you an example for this model, and then I'll briefly touch on these other broad themes that my research touches on, and then I will hand it over to Stefano. So just to give you a taste, a flavor of what our work is, let me show you this particular example. This is a molecule, it's a hexamer. There are 6 monomers that make up this hexamer, and each of these monomers can undergo the phosphorylation and dephosphorylation reactions that I had shown you earlier. In the futile cycle, we could phosphorylate and dephosphorylate the futile cycles. And in each of these monomers, that can happen twice. It can either have an orange phosphorylation or a pink phosphorylation. So here you see that this monomer is undergoing pink phosphorylation and then undergoing orange phosphorylation and then undergoes pink dephosphorylation and then orange dephosphorylation. So it's undergoing this futile cycle twice. And the numbers of the phosphorylation level are in the bracket. So the first number is the number of orange phosphorylations. The second number is the number of pink phosphorylations. And I just say this to explain the state space. And besides the phosphorylation level, you can see that at each given time, because each of these monomers can phosphorylate or dephosphorylate, that at any given phosphorylation level, the system has 4 choices to undergo phosphorylation in pink or orange or dephosphorylation in pink or orange. And what it's likely to do, what it's primed to do is given by these sub-labels A, B, C, D. And that forms a full state space. And so we can also look at the 4 internal states of the same phosphorylation level and allow for reactions between them that allows it to go to a different level of priming. All of this is a way in which all these reactions, it's a way to write down the full reaction space of the system. It's a pretty simple state, but yet the reaction space is quite large still. And so you can see that for each phosphorylation level, we get these 4 states that then tile the space of all the phosphorylation. So these are the 4 states for 0, 0. These are the 4 states for 1, 1 in this blue square. And you can see that going across horizontally, we are increasing in the orange phosphorylation. And in the vertical axis, we are increasing in the pink phosphorylation. This is a square lattice. And again, here now, one of our big contributions was to recast the language of chemical reactions as a sequence that can then allow us to exploit many of the results that have been developed for translationally invariant lattices. So here now, this can be recast as the lattice, and you can see that the internal states form a unit cell, and we can then use some of the tools developed for solid state systems, and especially lattices and periodic systems there. But now we can now use this in the appropriate language of biophysics to see and use the challenges of biophysics to extend these tools further, especially in this non-Hermitian dissipative regime. In particular, this lattice is very interesting because it has a phase transition between 2 different regimes. The first regime is the sort of boring vanilla regime, and that's diffusion. And you can see that because we have one cycle that goes, the black cycle goes clockwise and the gray cycle goes counterclockwise. You can see that there's roughly equal numbers of both cycles. And so the system will just sort of, when the black rates are roughly the same as these gray rates, the system will just wander through the whole space and will just ergodically cover the whole network. And that's just a random walk. That's just diffusion. But there is a phase transition when these black rates become larger than these gray rates. And in that system, the system will no longer undergo this slow random walk, but it will undergo ballistic motion on the edge of the lattice. And this is a hallmark of topology. And we can see this by inspection. So let's say we come down here, we wander down to the edge of the lattice. But once we go to the edge of the lattice, that you can see that the state space becomes very interesting. Here, we can only move forward. These black rates are faster than these gray rates. So now this is going to be more likely. This has to go here. This is more likely. This has to go here. This has to turn the corner. This is more likely. And it has to go here. And so now it's just going to go only around the edge of the lattice. And this is a topological property that instead of wandering in this whole space of the lattice, it is now only going on the boundary of the lattice. And so you can see that obviously the scaling is also very different. Instead of the volume, instead of the dynamics going in the volume, now it goes on the boundary of this lattice. And it is very intriguing and potentially very useful for biology, because biology often depends on dimensional reduction. So you've often seen biological networks that look very messy and very large, but so often, depending on the stimuli, on the environmental conditions, the system needs to take very specific pathways in order to achieve specific aims in a very short time. So it needs to move specifically through one pathway of the lattice in this dimensional reduction. And this is a big question across all biological networks. And so we are using these tools now to model this dimensional reduction and going on just the edge of the lattice. And not only does this system undergo dimensional reduction to go to the edge of the lattice, it also has a very nice property of robustness that topological systems have, which is that now if we have missing states, or let's say the system cannot reach certain states in the system, instead of having just this edge state hit a roadblock and then fall apart, here the system now, you can see it's going to hit this, reach this corner, it has to turn the corner. This is more likely now amongst these two rates. This is more likely, this will go here, this will have to go here, this is more likely. And so the system will always go around the edge of the lattice, even though no matter whether or not there's perturbations and there's deformations or there's missing states, the system will always just go around whatever edge of the lattice there is. And of course, we know that for any finite system, which all biochemical reactions are, there's a finite edge, the system will just always go around that edge and not be disrupted amidst deformations. So I know that this was just a 15-minute talk, so maybe I'll just briefly, how much time do I have left, just to get an idea?
I think 5 more minutes.
Okay, perfect, great. So that's perfect. So here we have the system that undergoes this dimensional reduction, but also its robustness has a property of robustness that is very attractive for biology, because here we can think in how development, as a system develops from being an embryo into a full organism, even despite different environmental conditions, the system can often pivot to stay on track and even adapt and react and continue its biological developmental trajectory. And this kind of robustness, or the fact that we can adjust very easily to changing temperature, changing environments, this kind of robustness is clearly essential for biological systems. And here we're excited to use this property of robustness for biological systems. And let me show you an example of what we've been modeling. So here you can see that the system can form a clock that goes back to itself, where the period is much longer than the underlying chemical reactions. So naturally, we were curious whether we could use this to model the circadian rhythm, or a long cycle, 24 hours, even though the underlying chemical reactions are much faster. And so here we have this hexamer. So now you see why my molecule had 6 monomers, and it undergoes, and this regulates the circadian rhythm of cyanobacteria, where each monomer has 2 phosphorylation sites, T and S. By day, all T sites get phosphorylated, and then all S sites get phosphorylated. And then by night, all T sites get dephosphorylated, and all S sites get dephosphorylated. Just to go through the biology, the biochemical space is still very large, even though with just these three proteins, this Chi-C, Chi-A, and Chi-B, and ATP in a test tube, the circadian rhythm can be replicated. So experimentally, it can be replicated very simply, but there's still a huge chemical reaction space. And the question is, why is this cycle so organized that it undergoes all T and then all S, and then T goes away and S goes away? Especially because we expect individual monomers to undergo individual phosphorylation and dephosphorylation. We expect monomers to bind into hexamers and unbind into monomers. Cryo-EM has even shown us different conformations in the same hexamer. So here we want to model the cycle, and we use our topological model to do so, where the orange and pink phosphorylations are now our T and S phosphorylations respectively. We can identify certain binding of ligands and changes in conformation to describe what these other chemical reactions could be. And now this is the same lattice, just that now we have the reverse rates for thermodynamic consistency. We have done an extensive comparison to experimental literature to show that this is consistent with what they see, including different priming reactions, as well as a difference in the black and the gray rates, because the black rates can be powered by consumption of ATP to be fast. And what we get is that here we get that our topological model will naturally show all T site phosphorylation, all S site phosphorylation on this molecule, and then all T site dephosphorylation and all S site dephosphorylation in this etch state in a topological phase. And this is achieved simply with a model that just has two parameters, the black rates and the gray rates. And just by tuning the ratio, we can get this topological state. And this comes about in this very natural ordering compared to many other models where, because of this precise timing where T has to turn on and then turn off, S has to turn on and then turn off, that here now, these models have many more parameters. And in fact, you can see that such models will often be a lot less robust because if you cut it here, the cycle will break. This is also just one phosphorylation, so the ordering, it doesn't come about naturally. I'm just going to close now by just mentioning that here we are also adapting a lot of results from quantum systems by mapping our master equation into the Schrödinger equation and using a lot of results in lattice systems. And we can also use simulations to verify our results, as well as look at other experimental platforms to test and to predict new properties, such as for self-assembly. I just want to close by saying that this is a really fun playground because now we have a new system, stochastic systems, that have many similar topological properties, but because now we're in the master equation, the formalism has also many important differences. And not only that, now here we are in the realm of non-hermiticity. These are dissipative, energy-consuming systems because that is the realm of biology, where the hawk grabs the fish and not the other way around. And so now here we have to develop new non-equilibrium tools. And we've also been developing a lot of fun theoretical tools. I'm not going to go into this because of time, but what we see here is that for the same network, the quantum system can have edge states, but not the stochastic system. And we need non-reciprocity to restore this. And we prove that this is true for any geometry and dimension. And overall, we're very excited to explore these systems more, both by looking at biological and stochastic applications are in synthetic systems and colloidal systems, as I showed you in my collaboration, as well as to develop new fundamental theory that can describe robustness in biological systems. And I would love to thank my group and my collaborators and all of you for listening. Thank you so much.
Thank you. So we have some minutes for questions. I don't know if there are any questions for Evelyn. Yes, there is one over there. Question from Lucilla de Archangelis.
Thank you.
Hi, Evelyn. Thank you for the beautiful work. I'm wondering, have you ever thought about the possible relation of your mechanism with the balance of excitation and inhibition that an healthy brain is able to realize when it's not in pathological condition?
That's very interesting because, yeah, the delicate balance of excitatory and inhibitory is very key, as you say, between healthy dynamics and disease dynamics. And we have not looked into this. I have also worked on neuroscience, and we are working to try to look at, what does robust dynamics mean in brain networks as well. And we haven't thought about excitatory and inhibitory, but you're right that would be an extremely interesting question to look at. Thank you for the suggestion.
Thank you.
Over there.
Thank you. That was a beautifully mind-blowing talk. I know that you're using random lattice theory, but it looks very much like a cellular automaton. Does it work? Are your models very much like a cellular automaton? And my other question is, glutamine synthetase, which is a 12-mer, has regulating factors. And it looks very much as though glutamine synthetase could behave like this.
Wow, thank you. So about cellular automatons, that is very interesting because that is also a system in which local rules can give very interesting macroscopic behavior. And I think what is very interesting about this interdisciplinary prize is that I think we are very much engaged in trying to look at connections between different disciplines. So it would indeed be interesting to see if similar principles could operate in those systems as what we are showing here. And thank you for telling me about this molecule. I would love to look more into it. I'm not familiar with it. So I would love to maybe email with you more about this molecule and see and learn from you why you think that it could connect. Thank you.
Are there any other questions? Over there. So the previous one was Igor Gledil, and now Prajpal Shastri.
Yeah, thanks for that. That was amazing. I was wondering if you're also able to predict the sort of plasticity in the circadian rhythm that can emerge depending on different environments, perhaps, or other factors?
Yeah, that's a great question. I have a student who's working on that now. So in our paper that was just published on Nature Communications, we did not look too much into plasticity mechanisms, although that is certainly very interesting and biophysically relevant. We were showing how this could be demonstrated in sort of the standard scenarios. But you're right that that is a great and very important question. And so I have a student who's working on that now, because you're right that questions such as temperature compensation, how is it that we would expect chemical reactions to change and proteins to change depending on temperature, and yet the circadian rhythm is clearly very, very stable to different temperatures. So questions like that, you're right, would be really fundamental to understand.
Any other questions? I have one question. There is no cooperativity here in the transitions, I think, because you said there were only 2 parameters, but still you see that some states are like preferred depending on something, on the prevalence of the 2 different types of loops that you get in that lattice. So what about cooperativity, which is usually, I mean, invoked to explain when you see some states more often than others?
Absolutely, and you're right. And so what I'm comparing it with is this typical cooperative paradigm, this Monod-Windman-Shanzhu paradigm. And what we are hoping is that our systems could provide an alternative mechanism to cooperativity, because cooperativity has clearly explained some things like hemoglobin very well. But for some other systems, there's still many mysteries, I think, that remain, where here, using a cooperative mechanism, it looks as if it is very difficult to account both for some of the experimental data where we see a lot more stochasticity, and yet we also see a very robust clock. And so we're hoping that our systems, our models can provide a different explanation that can account for both this stochasticity and this robustness at the same time. So potentially enriching our suite of available tools to maybe even provide a mechanism for what is usually thought of under the umbrella of cooperativity.
So what do you think? Because I was wondering many times when you have all these cooperativity indices that are so large, for example, for transcription factor binding to lead expression of genes. Do you think that these could explain why you get those cooperativity indices so high from when you see the data?
Yeah, so we are working on a project on gene transcription right now and looking to see how we can study these. Yeah, we are hoping that we can add some insights into the debate. Right now, we're still looking at smaller networks, but it is true that the wealth of behavior that we see there, it is very striking. And what we are very happy about is that our models can explain and predict these very striking switches and very striking transitions. As you see in our model, it can switch very quickly between diffusion to, moving around the edge. And in gene networks, clearly they switch on and off very quickly as well. So we are excited about that possibility.
Okay, thank you, Evelyn. So we thank Evelyn again.
Thank you all so much again.
And so we ask Stefano to share his presentation, please.
Okay. So first of all, sorry for my coarse voice. I'm recovering from a pretty bad cold. But so, well, first of all, I'd like to say that it's a great honor to receive this award, particularly from this organization. I have a great admiration for what IUPAP and its delegates do. I think the IUPAP's mission of facilitating international cooperation and standing up for physicists. Safety and freedom of expression is as important today as it's ever been. And with that, I'd like to tell you a little bit about work that I and very talented students and collaborators have done alongside me. So let me start by talking about what defines non-equilibrium. Sorry, I'm trying to hide the, okay. So what defines non-equilibrium? Well, so, there is one way of being in equilibrium, and there is infinitely many ways of being out of equilibrium. And so the best way of defining non-equilibrium is to actually define equilibrium. And you can find several definitions. And so here I pick one from the physical chemistry textbook by Kirkwood and Oppenheim, where they say that an equilibrium system is one in which intensive properties are independent of time and no current of matter or energy exists in the system's interior or at its boundary. And so now being out of equilibrium means that either of these conditions fails. But what we ask is there a quantifiable signature of how far from equilibrium a system is? And the answer, yes. If we look at this video by Edward Muybridge from 1878, this is one of the first films that has ever been recorded. And this is a video of a horse running forward on the left. And on the right, I played the same video backwards. And so now the horse, by moving forward and doing work, is clearly breaking time reversal symmetry. And that can be seen by the fact that if I play the video backwards, you can tell the difference, right? If the horse were doing no work and its motion was completely stochastic, then you wouldn't be able, as I'll show you in a second, then you wouldn't be able to differentiate the forward from the reverse trajectory. And the ratio of probabilities between the probability of observing a forward trajectory and its reverse, forward in time, is encoded by the fluctuation theorem that says that this ratio is equal to the exponential of a function, which is the entry production. And now if the system is out of equilibrium, this quantity, the entry production, is greater than 0, and therefore the probability of observing the forward trajectory is over the probability of observing the reverse in a video forward in time is greater than 1. And so entry production quantifies the departure from equilibrium. It quantifies the degree of time reversal symmetry breaking and the thermodynamic cost of maintaining the system out of equilibrium. So as I said a second ago, if I were to look at an equilibrium process, like for instance, the diffusion of fat globules in a glass of milk, now if I play the video forwards and I play backwards, the dynamics look identical because they're statistically identical. And so now the ratio of the probabilities is equal to 1. And consequently, entry production is 0 and the system is in equilibrium. So how can we go about quantifying this degree of time reversal symmetry breaking? Well, I've hinted at the fact that you can look at whether a forward trajectory is statistically similar to a reverse trajectory. So what we can do is that we can record a sequence forward in time, which we will observe with some probability P of X forward. And then we will reverse it, right? And so now in the forward in time video, we will observe the reverse trajectory with some probability P of X reverse. And one way in which you can relate two distributions is by using a statistical quantity called the Kullback-Leibler divergence. And the Kullback-Leibler divergence, from an information theoretic point of view, it's essentially the extra number of bits that you need to encode a sequence using a dictionary that comes from a different distribution. Okay, so if the dictionary that you're using is distributed the same as the forward as the signal that you're trying to encode, then there's going to be no extra information and the Kullback-Leibler divergence will be 0. But if you're using a dictionary that is suboptimal because it belongs to a different distribution, then you're going to need extra bits of information and the Kullback-Leibler divergence will be greater than 0. And so one way in which you can write this is as a difference between a cross entropy between the forward and the reverse probabilities minus the entropy. And remember this because I'm going to use it in a second. Now, it can be shown that the Kullback-Leibler divergence is just the entry production. So the way we quantify the entry production is by introducing an estimator that is based on data compression. And the way it works is as follows. We look at a sequence forward in time, and then we split it in half, okay? So then we pick a random, element of the second half of the sequence, and we look for the longest match anywhere in the first half of the sequence. And it turns out that now, because they're both forward, okay, if I try to compress the second half with the first half, I'm gonna get the entropy. Okay, and so you can basically, if you measure the average length of this match and you compute the logarithm of the length of the sequence divided by the average length of the match, you get an estimator for the entropy. So then what we do is that we take the first half and we flip it. Okay, so now if the first half is being flipped and is not statistically equivalent to the second half, it's going to be harder to compress the second half. And so again, I pick a point on the second half, I look for the longest match. And again, now the log n divided by the average length of these matches is the cross entropy between the forward and the reverse trajectory. And their difference is the KL divergence. And this particular estimator that we introduced outperforms all non-estimators of this class. If you take a three-state macro process, there are at least 3 orders of magnitude more efficient than alternatives that have been proposed prior to this approach. And so now that we have a method that allows us to efficiently quantify time reversal symmetry breaking, let's apply it to something interesting. So I was a postdoc in Paul Chaikin group, and he was, he's an experimentalist, I'm a theorist, but I was very fascinated by experiments that they were doing at the time. So they were studying this process called motility-induced phase separation. And the idea is that if you take TPM particles, they embed an hematite block, and now you put them in a nitrogen peroxide solution and you shine blue light on the system, what will happen is that these colloids will start self-propelling. And now something really surprising happens, which is that these colloids cluster together, even though there is no cohesive interaction in the system. And the way you can see there is no cohesive interaction is that if I turn the light off, so let's keep forward, now we turn the light off and everything falls apart. And so you can see that the clustering is induced by the mobility of the particle, by the self-propulsions of the particles, and the fact that the mobility now is dependent on the environment and the local density. Okay, so Michael Cates and co-workers were developing statistical field theories to try and understand this phenomena at a coarse-grained level. And so the way they approached the problem was by introducing a Cahn-Hillier type model. Okay, so we just have your continuity equation where you have a flux, which is the gradient of a chemical potential. But now what Mike and collaborators did was that they took the standard equilibrium free energy functional and they added the lowest order correction that you cannot derive from an equilibrium free energy functional. And so now if you do that at the level of the density, it just looks like a standard Cahn-Hillier model. But if you look at the dissipation in the system and at the entry production locally at every pixel, you see that the system is essentially equilibrium everywhere. You have no dissipation everywhere except at the interface, okay? And this is, of course, a coarse-grained result. It means that at this coarse-grained scale, the system is equilibrium pretty much everywhere except at the interface. And I wanted to see this in an actual system. I wanted to come up with an approach to actually measure this departure from equilibrium on some scale in a real system, not in a field theory. And so what we did is that we took a simulation of active Brownian particles. We discretized our system here. The grid size is exaggerated. In reality, we use a grid that is about the size of a particle, as I'll show in a second. And now what we do is that at every location in space, we track the occupancy of these 2x2 blocks, okay? So basically, I can have 5 possible states, right? You know, either one of the possible positions is occupied or none of them is occupied. And I track the time series of the occupancy of this 2x2 block as a function of time. And then I compute the Kuba-Leibler divergence between the forward trajectory and the reverse trajectory, just like I described at the beginning of the talk. And if I do that, I get exactly what was predicted from the field theory, which is that the system is actually the most out of equilibrium at the interface, because that is where I measure the largest Kuba-Leibler divergence and therefore the largest entry production. And at this scale, everywhere else, the system is essentially in equilibrium, okay? Now, why is this happening? Well, the reason why this is happening is that when the self-propelled particle is approaching the interface forward in time, what will happen is that it will approach the interface and then it will slowly rotationally diffuse until it will follow the interface of the cluster, okay? If I play this backwards in time, I see something that I will never see forward in time, which is a particle moving along the interface and then spontaneously stopping and turning away from the interface. That almost never happens. And so what will happen in reality is that if a particle is diffusing along the interface, it will just keep going, okay? And so this is where the time reversal symmetry breaking is happening near the interface, and that is why we're, the picture is lighting up at the interface of the big cluster. Now, of course, you can look at larger scales, and so instead of taking a 2x2 block, you can look at a 3x3 block or a 4x4 block. And now what happens here is that you start picking up different modality of time reversal symmetry breaking. In fact, if now I can accommodate two particles in my block, I can pick up collisions between particles, and obviously collision break time reversal symmetry, because if two self-propelled particles bump into each other, they will slightly rotate past each other and take off. But if I were to play backwards, basically reverse their velocity, they would just run past each other, right? And so each collision is a time reversal symmetry breaking event. And if I now can pick up three particles, I'm going to see even more modes of time reversal symmetry breaking. But the main point is that the majority of the introduction is always at the interface, regardless of how many degrees of freedom you're tracking.
You have two more minutes. You have 2 more minutes.
Yeah. Okay.
Yep.
I have 2 more minutes. Okay. Well, so you can actually relate this to a work extraction mechanism. And the idea is that when I measure the local entry production, I can ask whether I can extract work from it. And we by non-equilibrium linear response, we derive a relationship where we basically show that the power that can be extracted is only non-zero if the entry production is non-zero. And in fact, if I only track the position, it's the same as coupling to the positions of say this bullet that moves along this track. And that is like putting a turnstile that turns whether the particle goes from I to I+1 or in reverse. But if I track the orientation, now I can differentiate forward from backwards. And now it's like coupling a mechanism that only turns in one direction. And indeed, now I have positive entry production, and I can actually extract some power from it. And so we've characterized this in a system of swimming bacteria in a microfluidic chamber, which is an experimental system. And we wanted to understand this a little bit better. So what is happening here is that the time reversal symmetry breaking of the bacterial motion is coupling to the symmetry breaking in the underlying geometry of the chamber. And so, if these particles were equilibrium particles, you would have a constant concentration across the two sides of the chamber, but because they are breaking time reversal symmetry, you end up with higher concentration on the right than on the left. And to understand that, we actually did experiments in collaboration with Shang Chen. One of my students did the experiments, and we constructed these beautiful PDMS microfluidic chambers where you could visualize in detail what the bacteria are doing. And we built a microscopic model, which, despite the dozens of work on this type of systems hadn't been done. And what we were able to show is that there is only 3 factors controlling bacterial rectification in such a system, which is a universal distribution of self-propulsion angles at the gate, a realignment along the solid boundaries, and bacterial wobbling because E. coli actually swim in an helical trajectory. And by using these ideas, we were able to build a parameter-free model that predicts experimental and simulation result. And it tells us that the highest efficiency is around 120 degrees. So now let me close with an observation, which is that active rectification is actually leveraged by carnivorous plant. So the Genlycia is a carnivorous plant that uses these subterranean leaf structures called rhizophils to feed on microorganisms on the soil. And if you do ACM of the interior of these rhizophils, there is hair which are about 45 to 70 degree half angle. And that is completely consistent with the 60 degrees that we predicted to be the most efficient. And so what this seems to suggest is that the plant may have actually evolved to have these optimal structures so that they could pump bacteria as efficiently as possible through the subterranean leaves into the vesicles that are responsible for digestion.
So your time is sort of up.
Yeah. So I'm gonna conclude, yeah, I just want to thank the students and collaborators and funding agency. Thank you.
Thank you.
Oops. Oh yeah, just I'll leave it here.
So, are there any questions? So, oh, over there.
Thank you, Stefano, for the nice presentation. I'm Quack from Singapore. I was wondering, quantum mechanics is actually time reversal, at least for the pure state case. Of course, you can make it time irreversible with master equation. But what is interesting perhaps is that unitary evolution typically involves a cycle. So it depends on how you reverse your cycle. So you need to at least know what's the period in that time reversal. Could you comment on that?
Well, I mean, the quantum mechanics is time reversal because it's unitary, right? So I just run it forward and run it backwards and nothing changes. You can't really differentiate. In these systems, you can because you have an actual orientation, right? So you can tell the head from the tail of the bacterium. So if I play a video forwards and I play backwards, I now see the bacterium moving in the direction of its tail, which obviously it's not something that I would observe forward in time, right? The key of this measurement is that the time reversal symmetry breaking really depends on the degrees of freedom that you're tracking. So you could have indeed a system that is out of equilibrium, but given the degrees of freedom that you're observing, it looks completely in equilibrium, right? There was, for instance, in the example where we looked at this local entry production for the particles, if we don't track sufficient degrees of freedom to catch the collisions, we will conclude that the low density phase is completely in equilibrium, right? And so, whether you're in whether you detect the non-equilibrium nature of the process depends on whether you have selected the right degrees of freedom.
Any other questions? I have, over there, Rajpal.
Yeah, thank you for that amazing talk. You showed in the earlier part of your talk that when you shined blue light on colloidal formation, the formation kind of fell apart. Given your analysis in the latter part of the talk, what exactly is happening here? I didn't quite. So how do you explain the physics here?
Oh, how do you explain the motility-induced phase separation?
Yeah, how do you
So,
explain
yeah.
what you saw here with the, when you shine the
Well,
blue
what is
light?
happening is that the particles are persistent. And so when they start pushing against a cluster, they maintain the same orientation. And so now your mobility depends on the local density. So what's happening is that in the vicinity of a cluster, you have much lower mobility than away from a cluster. And so that basically you get into this arrested state when you have a fluctuation that leads to an increase in local concentration. And so then that leads to a clustering event. That is the explanation for the motility-induced phase separation.
So then that wouldn't happen if they are self-propelled particles.
This only happens if they're self-propelled. When they are not self-propelled, this doesn't happen because there is no persistence.
Okay, thank you.
Okay, we thank Stefano again. And we congratulate both Evelyn and Stefano for their great work, the very interesting talks, and for the award, of course. Thank you.
Thank you very much.
So we will end this Zoom, go to the coffee break or tea break, and come back to the old Zoom. Thank you. Thank you.
So we will come back at 10:35. So on the Indigo Zoom room, we come back here at 10:35, which means in 20 minutes.
边